Hi, my name is Mandi. I'm a postdoc from Stanford University, and the title of my talk is Finding a Cure for Colon Cancer. Colon cancer is the second leading cause of cancer-related deaths in the United States. There are currently no cures. Over 95% of patients have mutations in what is called the Wnt pathway, which is a cell-to-cell communication pathway that controls when cells grow and proliferate. The mutations that these patients have cause this pathway to be overactive, and this results in uncontrolled cell growth that leads to cancer. We know that overactive Wnt signaling is the major cause of colon cancer. It drives both the initiation and the continued progression of colon cancer. However, we also know that Wnt signaling is a very important part of normal tissue maintenance in many organs. So we can't just simply inhibit the Wnt pathway altogether to treat colon cancer. Even though that will treat colon cancer, that will also destroy other tissues and organs in the body. So we need to identify vulnerabilities that are specific to the cancer-causing overactive Wnt pathway that do not harm these other tissues. This is the key to developing viable therapeutics for colon cancer. The majority of mutations that cause overactive Wnt signaling are found in a single tumor suppressor gene, and this gene is abbreviated APC. APC normally plays a key role in keeping the Wnt pathway in check, thereby preventing improper cell proliferation. In colon cancer patients that have mutations in APC, the Wnt pathway goes unchecked, and this results in uncontrolled cell growth that leads to tumor formation. What is puzzling, however, is that these mutations in APC do not completely destroy the function of APC. Rather, they just seem to dampen its functionality. And we don't fully understand why this dampened, partially functional form of APC is really effective in driving tumor formation. But we do know that if the full functionality of APC was restored, this would be enough to stop tumor growth and even cause regression of existing tumors. And this is seen even for tumors that contain other cancer-causing mutations, meaning that if we could restore full APC function or find another way to tune down only the cancer-causing Wnt pathway in these patients, we could potentially cure colon cancer. So in order to find vulnerabilities that are specific only to the cancer-causing Wnt pathway, I began by asking a fundamental question. What are the genes required for Wnt signaling when APC is partially functional? And remember, this is the cancer-causing form of APC. So this gives us extremely valuable information because we want to understand how overactive Wnt signaling is different from normal Wnt signaling and which genes account for these differences. We can then target these differences to inhibit only overactive Wnt signaling, and in this way, targeting only the cancerous tissue without harming healthy tissue. In parallel, I will also use biochemistry and protein engineering strategies to understand how and why mutations in APC cause partial functionality, and which functionality or functionalities are lost. Our hope is that because the cancer-causing APC is still partially functional, we can figure out a way to enhance that function to stop cancer progression. And so this requires a very detailed understanding of the other proteins that APC interacts with and exactly what types of interactions are lost or diminished. And once we have a better picture of what mutated APC is lacking, we can then find ways to try and fix it. In order to find a cure for colon cancer, we first need to understand the differences between the overactive Wnt pathway that causes colon cancer and the normal Wnt pathway that healthy organs depend on for normal function. We also need to have a more detailed understanding of how mutations in APC drive tumor formation. Our whole philosophy for this project is to find vulnerabilities specific to the cancer-causing Wnt pathway that don't interfere with normal Wnt signaling. We can then exploit this as a powerful therapeutic for colon cancer, as well as other cancers that are driven by overactive Wnt signaling.